And a warm welcome to our regulars, our Garvan family members, our donors, partners for the future, corporate supporters. We thank you for joining us. Today is our first public seminar of the year, and I think we've got a fantastic program looking at genomics and inherited disease. And we've got two incredible speakers for you, Associate Professor Owen Siggs, who is co-director of our Genomics and Inherited Disease program here at Garvan, a large Garvan venture combining a large number of laboratories here focused on unpacking and being the detectives of Garvan to understand the underlying cause of inherited disease, which many of you may know, whilst we call them rare or inherited diseases, they actually affect more than 8% of the population. So I think we can all agree that's not exactly rare. We've also got Dr. Amali Mallawaarachchi, who is one of our clinician scientists, a senior lead within the inherited kidney disease laboratory here at Garvan, with our former executive director, Professor John Shine, who some of you will know. Now, in terms of the program, we're going to hear from our speakers, and then we are going to open up for a Q&A, and we will accept questions from the audience and also from the Zoom room. So if you have any ideas throughout today's session, please do grab a pen if you've got one and note down the questions that you'd like to ask. It's now my great pleasure to welcome Associate Professor Owen Siggs. Thanks, Owen.
Thank you, Mara-Jean. Fantastic to see so many new faces in the room and so many names online. I realise, though, that many of you will have no idea, certainly who I am, what Garvan is or what we do here. So I just wanted to spend the next 15 or 20 minutes or so just introducing you to those three things. And I wanted to start with a story about a patient. So this patient, a young boy, about 10 years old, one day he was out playing basketball in the schoolyard, cops a ball on the back of the head. Quite painful at the time, goes home and talks to mum and dad over the dinner table, sees the doctor the next day, and the doctor organises a CT scan. And on the CT scan, as you'll see, there's a lump at the back of the head and it's eroding into the bone. So not quite sure what that is, so they refer the young boy to the surgeons, and a few weeks later the neurosurgeons cut out this lump, send it to the pathologist who put it under the microscope, and they see this. And you might notice this sort of river of pale cells through the middle there. They also put it under an even stronger microscope, looking deep inside the cell, and saw these unusual structures that look a bit like a 1950s wooden tennis racket. Both of these things characteristic of a disease called histiocytosis X. And like I'm sure many of you in the room, this is something that you've never heard of. But this patient was me. My family and I, we're talking to the surgeons afterwards, and we had no idea what was going on. We were told this was a 1 in 1 million disease, so extremely rare. But at the time, 1 in every 4 children who was diagnosed with this disease would die within 5 years of diagnosis. So quite a frightening experience at the time, but fortunately for me, this was just a single isolated lump, and so the operation was curative. But a lot of other children were not so lucky. Fast forward 30 years now to today, and we now know an awful lot more about this disease. Most importantly, we know that it's a genetic disease. So you'll hear a lot about these this morning, but these are caused by, in this case, a single change in our genetic code. And not only that, but that genetic discovery pointed directly to a drug, a tablet you can take twice a day that blocks the effect of that genetic change. And some of the first trials of this drug were truly spectacular. So like this young girl down the bottom here, this drug just melts away these 2 big lumps she has in her neck within just 2 weeks. Or in this man who has a similar disease spread throughout his bones primarily, and you don't need to be a radiologist to appreciate how these black areas of disease activity in the bones just start to disappear over time. First after 1 month of treatment, and by 4 months of treatment, almost vanished. So in quite a short space of time, this disease had gone from being something that was lethal in 25% of patients when I was diagnosed to something that can be treated today with a tablet. And all of that was thanks to medical research happening in buildings like the one you're in now. And as it turns out, this is now exactly what I do for a living. 2 simple steps: understand what causes disease and then use that information to improve how we manage it in the clinic. So this is the journey that brought me to Garvan. I grew up in Adelaide and studied science and a medical degree there. In fact, my very first university lecture was the same day that the first draft of the human genome was published. And I've been hooked ever since. I then went on to Canberra and Melbourne to train in some of the other top medical research institutes in the country. And then after that, I decided to go overseas to do a PhD, and I did that PhD between Oxford in the UK and La Jolla in Southern California, where I trained with a Nobel Prize winner. After that, I moved to the US and the UK to train in 2 of the top genomics institutes in the world. And then in 2021, moved to the Garvan Institute to set up a lab. And Garvan really is the premier genomics research institute in Australia. So what is Garvan? Well, we're a bit over 60 years old now. We've spun out of St Vincent's Hospital across the road, and we're a group of about 600 researchers or support staff working on more than 50 diseases or disease areas. And importantly, we're funded by the public, either indirectly through grants from governments and charities or directly through donations. And we work for the public. So we work on some of the key unmet needs in health and medicine where we believe that we can make the biggest impact. And the way that we do that is really through the genome. And genome and genomics is at the core of everything we do. So it's also part of our mission to use the information encoded in our genome to better diagnose, treat, and prevent disease. And the beauty of genomics is that we can use it to understand just about any disease. Whether you've got cancer or MS or histiocytosis X, genomics is transforming the way that we understand and manage all of these diseases. So what is a genome? Well, it's our own unique blueprint. It's a set of instructions that build and maintains our body. It's present in almost every one of the 30 trillion cells we have in our body, a 6 billion letter code stretched across 2 meters of DNA, and then that's crammed into 23 pairs of chromosomes, half from our mothers and half from our fathers. And then spread across these chromosomes are 20,000 genes, each of which encodes a unique protein building block. Now, all of our genomes are subtly different, so on average, you and I differ in our genomes about once in every 1,000 letters, and most of these changes are completely harmless. Some of them are actually beneficial, but occasionally all it takes is one of these changes to go from someone who's perfectly healthy to someone who's very, very sick. Sometimes that's something you're born with or you inherit from your mum or your dad, or sometimes it's something that you pick up during your lifetime, like it did in my case. And just having this information can be incredibly useful, not just for the research we do here, but it's now permeating into almost every branch of medicine, from conception all the way through to death. So during pregnancy, for example, we now routinely use genetic testing to detect the changes that might cause Down syndrome in a pregnant mother. Even before a child is conceived, prospective parents can now test themselves to see if their future children might be at risk of severe diseases like cystic fibrosis or spinal muscular atrophy. And as we get older, there are more and more applications for genomics in medicine, a lot of which were, really were science fiction 20 years ago when I started in this field. And there are plenty more really, really exciting applications that are on the near and long-term horizon. But let me tell you about an area where genomics is really having a transformative impact in medicine right now, and that's in the diagnosis and management of genetic disease. So I'll take another case example now to illustrate this point. So this time, a story about a girl called Carla. When Carla was 3 years old, she was diagnosed with a very severe and crippling form of arthritis. So in her case, a really nasty disease affecting her hands and her feet in particular, making it painful for her to play, to walk, or even to draw. and totally unresponsive to treatment. So despite all the time she spent at Westmead Kids Hospital receiving it. A couple of years later, Carla was referred to a research study that we had running at the Garvan by her rheumatologist. And after sequencing her genome, we quite quickly found a mutation in one of her genes called CTLA4, which was causing her immune system to attack her joints. So this information was useful by itself. It provided an answer as to the cause of her disease, an answer to the family and to her clinical treatment team. But the most important thing for Carla was that this also pointed directly to a new drug, and a drug that could potentially work in her case. This drug was called abatacept, and the beauty of this drug is that it was a direct substitute for the genetic change that Carla had. So we got in touch with her rheumatologist straight away, and within a few days had approval to start Carla on this treatment. And within a few weeks, Carla was a completely different person. So going from a child like this, in and out of hospital, unable to do the sorts of things that a young child would take for granted, to someone like this, bouncing on trampolines, riding bicycles, even being able to draw. So this is really exactly the sort of thing that makes this job so exciting, being able to take a new technology to understand disease and then using that information to have a transformative impact on someone's life. Now, there are literally thousands of diseases out there like Carla's, caused by a single change in a single gene. And as Mallawaarachchi mentioned before, they might be rare individually, but collectively, they're extremely common. So there are hundreds of millions of people out there worldwide affected by these diseases, including me, and including many of you in the room, whether you realise it yet or not. Finding the genetic cause of these diseases can be truly life-changing for a lot of people, or they can at least bring us one step closer to a treatment or a cure. But despite all the progress we've made in genomics over the last 20 years, most rare disease patients still don't have a genetic diagnosis. And even the ones who do, less than 10% of them have an effective targeted therapy, like they do now for Carla's disease or for mine. But fortunately, these are exactly the types of problems that medical research can solve. And these are some of the faces of the people that are working on solving these issues at Garvan. So we are the Genomics Inherited Disease Program, a critical mass of like-minded researchers and clinicians. We've got specialist expertise across a range of different technologies and disease areas. In my team, for example, we work on diseases that cause inflammation or blindness, but we've also got experts in diseases affecting the kidneys, the brain, the heart, and the immune system, a whole range of different disease areas. So as well as working very closely with one another, we also work together with patients and their clinicians, with researchers, and with companies around the world. So this really is an international team sport. And in terms of what we're actually doing upstairs and next door, it all begins with patients with a known or suspected genetic disease, like Carla, with a special focus on those patients, the 50% or more, who despite all the best efforts of the healthcare system still don't have a genetic diagnosis. And the next step then is to take samples from these patients and apply the latest and very best technologies to try to find a diagnosis. So that might be a new sequencing instrument, or it might be a new machine learning method that we're seeing more and more of every day. And this part is the real needle in the haystack operation. So a lot of computational heavy lifting and careful analysis to find sometimes that one single change amongst 6 billion letters. And then finally, for patients who do have a genetic diagnosis, we're here to help them get the very best outcomes. So in some cases, like Carla, there's a clear targeted therapy. In others, there might be a clinical trial or another treatment that's on the horizon. Or in some others, we might still need to do a little bit more research and development to get there. But this last phase is a really, really fast-moving field. So largely thanks to advances in genetic therapies from companies like Moderna, that you might have heard of from COVID vaccination technology. The cost of developing these targeted therapies now has simply plummeted. So there's an absolute tsunami of these specific genetic medicines coming through the pipeline. So we really need to work as quickly as possible to find the right patients and match them to these treatments that are on the horizon, and get benefit as quickly as we can. But let me come back to the patient. So they're the most important part of this process, and they're the foundation of everything that we do. Most of them don't have a diagnosis or a treatment, or even a dedicated charity to help them find one. Some of them are so rare there might only be one or a couple of patients across the whole country. They're scattered across states, across cities, across hospitals, and most of them don't have an easy way to get access to the best medical research. So that's something that we really wanted to change. So as part of our program at Garvan, we've launched a new national study to make this whole process a lot simpler for both patients and researchers. And what makes this study unique is the way that patients and their families can participate. So you don't need to attend a specialist hospital or clinic, no matter whether you're living in Darlinghurst or Darwin or anywhere in between. If you have an eligible disease and you can complete an online questionnaire, you can join. So from this point onwards, after participants join, we act as a concierge. So we find the right researchers, either at Garvan or beyond, who might be able to help. And the response so far has really been incredible, so we're very excited to be underway and hope to have more success stories to share with you in the coming years. Just before I finish then, I wanted to give you a sense of just how fast this field is moving. And the best way to explain this, I find, is with this graph. So it's the cost in thousands of US dollars of sequencing a human genome over the last 20 years or so. So 20 years ago, when I got into this field, it cost 100 million US dollars to sequence just one single human genome. And that was something that only really huge international consortia could afford. Today, we can sequence a human genome for less than $1,000. And later this year, it's probably going to come down to 100 US dollars. So we're rapidly approaching the point at which it's going to cost more money to post your sample to the lab than it will to actually sequence it. So because of this, also the volume of genomic data is absolutely exploding. So by one estimate, we'll be generating next year almost 20 times more data than YouTube. That's a massive challenge for us as a field, but it's also a huge opportunity. And we're already seeing some incredibly powerful AI models being trained on these data sets. And those in turn are making this genomic information more and more and more useful in the clinic. And one of these opportunities is not just being able to diagnose a disease after it happens, but potentially being able to predict who is at highest risk of a disease before they even develop symptoms. So this is the space that's got the potential to have an absolutely enormous impact in terms of both the number of people who could benefit from this, but also the mortality and the morbidity that we could avoid by treating so many of these diseases earlier. And in many cases, we already have fantastic treatments for these diseases. So one example I wanted to give you was a disease, glaucoma. So in my clinical life, I treat a lot of people with this condition. It's a neurodegenerative disease that affects the optic nerve and vision. It's a really tricky disease for a number of reasons. It's also quite common. 3% of Australians over the age of 50 have it. Once you lose vision from glaucoma, you can't get it back. It's also asymptomatic in the early stages. So about half of people with the disease don't realise they have it. But most importantly, if we can detect it, there's plenty of fantastic treatments out there available to treat it. So we really desperately need new ways to find people as early as possible in this disease process so we can treat them and prevent blindness. And we think genomics is a fantastic way of doing this. So together with a group of colleagues, we've now developed a genetic test that helps us predict who is at highest risk of getting this blinding disease before it's too late. We then use that information to make sure that the highest risk people get seen earlier and more often, and even treated earlier and more aggressively. So these types of tests already available in the clinic, but it's also an approach that we can apply across a whole range of different common adult onset diseases like cancer, cardiovascular disease, all using information that we already have sitting right here in our genome. So let me leave it there. I hope that's given you all a useful taste of the type of research that we're doing at Garvan here today, and also the potential impact that it has to change patients' lives tomorrow. So I'll leave it there. very happy to take questions at the end or after the seminar. I'd also now like to introduce my colleague Amali. So Amali is one of our rising stars at Garvan. She's been here since 2015 when she joined as a PhD student, and she's also Australia's first and only dual-trained specialist in clinical genetics and kidney disease. So Amali's got some fantastic work to present to you today and really having a real immediate clinical impact. So thank you, Amali.
Hi, everyone. It's a pleasure to be here today, and thank you, Owen, for the kind introduction. Today, I'm going to speak a little bit about work that we do here at the Garvan through the Genomics of Inherited Kidney Diseases laboratory. And so I'm going to talk a little bit about genetics and kidney disease, and particularly focus on a condition called autosomal dominant polycystic kidney disease, which is one of the main conditions that we focus on here in our laboratory and the most common genetic cause of kidney disease. And so we'll talk a bit about research that's happening right here at Garvan in that condition. So kidney disease is really common. It's actually super common. It affects about 1 in 10 Australians. And when we think about kidney disease, I think we often think about conditions like diabetes or high blood pressure, and they're certainly really common causes of kidney disease, but actually genetic causes of kidney disease also make up a significant proportion of chronic kidney disease in Australia. In adults, about 10% of adults with chronic kidney disease have their disease because of a genetic condition. And in children, it's much, much higher. About half of children with chronic kidney disease have kidney disease because of a genetic cause. And we know that about 30% of patients with kidney failure, so who are on dialysis or who've needed to have a kidney transplant, have a family history of kidney disease. You can't come to a Garvan seminar without getting a little bit of a biology lesson. And so just to make sure we're all on the same page about genetics before I go further, I just wanted to talk a little bit about some genetic background. So I guess we all know that our body is made up of millions and millions of cells. And in the middle of our cells is our nucleus, which contains our DNA, which is the instructions to make all the bits of our body. When we have a look at the DNA under the microscope, this is what it looks like. And these sticks are called chromosomes, and they come in pairs because we get one from mum and one from dad, so that we're 50-50 of our parents. If we have a look at our chromosomes in even more detail and we kind of unravel them, they're made up of tiny little components that we call genes. And our genes are the instructions for one really specific function within the cell. And when we have a look at our genes in even more detail, they're a code, a bit like a computer code, a really fancy code that our body decodes to make things, to make proteins that are the building blocks of our body. And in patients who have genetic disease, they have a difference in that code, a tiny spelling mistake somewhere that results in particular conditions. And in genomics, a lot of the work we do is to have a look at that code in a lot of detail and try to decipher where there are differences that are causing changes or disease. In our laboratory, we focus largely on a condition called autosomal dominant polycystic kidney disease, which I call ADPKD for short, because it's a bit of a mouthful otherwise. It's the most common cause of genetic kidney disease. It affects about 1 in 1,000 people, so it's a pretty common condition. And in this condition, what happens is that little fluid-filled little cysts overcome the normal kidney tissue. And what you can see here is that there's the much bigger kidney there is a kidney of a patient who's got polycystic kidney disease, and you can see all those bubbles are little fluid-filled cysts. On the other side is a normal kidney, so that's what our kidney is supposed to look like. Those cysts take over the normal kidney tissue so that there's no working tissue left to do the work, the filtering of the kidney. And so it leads to kidney failure. And for about half of people who have this condition, they need dialysis or a kidney transplant by the time they're in their 60s. This is a familial condition, which means that if you have this condition, each child that you have has a 50% chance of inheriting the same condition. And we know that ADPKD is mostly caused by a spelling mistake or changes in one of two genes that are called PKD1 or PKD2. And one of the things that I find really remarkable is that despite this being one of the most common types of genetic disease, most families who have ADPKD do not know the specific genetic change that causes their disease. And there's a number of different reasons for that. And one of them for many, many years has been that technically it's quite difficult to read the DNA sequence in these genes. And that has made it very difficult to actually diagnose this condition genetically. But why does this matter? Why does understanding genetics matter? You could see how extremely abnormal that kidney looked in that person with polycystic disease. And so you can use ultrasound and other things at moderate stages of disease to diagnose this condition. So why do we care about the genetics? The genetics gives us extra information. It gives us information much earlier in the piece so that people can know much earlier that they're affected with this condition. And it helps us to give much more directed and guided care to patients with genetic conditions. Some of the most common questions that I get asked in my clinic is, I have this condition, am I going to pass it on to my children? And are there ways that I can avoid passing it on? This is a picture of a family tree of a family with polycystic kidney disease. And this is what family trees of most people with ADPKD look like. The coloured in purple squares and circles are men and women who have the condition. And the different layers there are different generations. And you can see across every generation, there's a number of people in this family who have this disease. And what patients with PKD want to know is the next generation going to inherit this? And for people in the family who appear not to have the condition, they want to know, do I definitely not have this condition? Can I give my kidney to my brother or sister who needs it? And so those are the kinds of questions that genetics can answer. Previous work in our laboratory here at Garvan used new, or at that time, new DNA sequencing technologies to develop the first genetic test for ADPKD in Australia. And before that time, there was no way in the research world or in hospital laboratories to do genetic testing for ADPKD. And we're really excited that after that first initial experiment, there's been some amazingly rapid progress in that space in Australia. That initial study that we showed developing that new test was in 2016. And today, which is not very far since then in the medical world, there's testing available in a number of different laboratories across Australia for Australian families with this condition. And it's funded through Medicare, which means that a lot more families with this disease can now have the opportunity to get a genetic diagnosis and use that to manage them better. And that original work is now providing the building blocks for the future or the research that we're working on in the laboratory now. That initial work has led to most patients with this condition being able to receive a diagnosis fairly straightforwardly. And so we know that that original test can diagnose about 90% of families with ADPKD. But what we're interested in is the small part of the pie of patients who, even after that type of genetic testing, remain without a diagnosis, which means that those families can't get that more personalised care that a genetic diagnosis allows. And so what our laboratory has been doing is collecting patients who didn't get a diagnosis from the original research studies that we collected, and then also through collaborations with genetics clinics and kidney units across Australia, seeking out other patients who've also not been able to have a diagnosis, and grouping all those patients together and using more detailed genetic sequencing technology and methods to try and understand what's causing the disease in those families. And what we found when we did that combined analysis was that most of those patients did have a genetic cause we could identify. It was just outside of the areas that we normally look at. This is a pretty complicated picture, but what it's showing is a picture of the PKD1 gene, and the blue rectangles of different thickness are the parts of the gene that we tend to focus on, both in the hospital world and in the research world as well, because that was our kind of basic understanding that that's where most genetic changes that cause disease happen. But what we actually found in these patients who were previously undiagnosed was that the changes in them were actually not in those areas. They were in the lines in between, the areas that we tended not to focus on as much. And these patients had changes in those areas that were causing their PKD. And what this has led to is to say, well, maybe we need to be extending the way that we do our analysis generally to be able to identify these types of changes. And this has really come about because of the power of numbers. And I think it nicely reflects some of the amazing potential of the registry that Professor Siggs was speaking about in his talk, that we were able to make these discoveries more straightforwardly because we were able to group together different patients who all had a similar common problem in that they had PKD and they didn't have a genetic answer. And when we looked at those patients together, the data was much more powerful to be able to find an answer in them. And that is the basic way that the registry works, that it allows us to group patients with particular conditions or particular genetic changes and say, let's focus on these as a group and see if we can identify ways to look after these patients better and to diagnose them better. And in rare disease, that grouping of patients or cohorting of patients is a very powerful way to make leaps in terms of our understanding and discovery. And so our current research continues to focus on these undiagnosed patients and continues to apply even newer genetic sequencing technologies that Garvan has here under this roof to try and better understand PKD. And these are some pictures of DNA sequences using something called long read sequencing, which means that we can have a look at DNA in really long pieces that allows us to understand things about structure of DNA that we never used to be able to do previously. And so I'm going to go back to this question about why all this matters. As a clinician who goes back to see patients in the clinic and to give answers to individual families who are incredibly affected by these rare conditions, it really matters to the individuals involved who can finally have an understanding or a very black and white reason why they have a particular condition that affects them. It never ceases to amaze me when we give back reports to families to say, it was this specific spelling mistake that caused the condition in you or your child. How powerful that is, that most people stop and say, okay, so this is really genetic. It wasn't something I did. It wasn't something I ate in the pregnancy. It wasn't a medicine I forgot to take. This wasn't my fault. And that in itself is extremely powerful. All of this work also improves the diagnostic rates for families in the future. The work we did in that small group of undiagnosed patients now means that future people won't be missed because of those differences that we didn't initially understand. And so it improves our testing and it improves our diagnostic technology. And I think equally importantly is that when we look and we deep dive into these families with really specific, particular undiagnosed conditions, we often then improve our knowledge base, and it's those things that eventually lead to cures, which is what we're all here to try and achieve. So I'm going to stop now and just say thank you to many, many people that I work with, because science is always a team effort, and also to many generous funders that we have through grants and through very generous bequests that make all of this research happen, and of course to the patients and their families who very generously participate in the research that we do here. And I look forward to hearing all of your questions. Thank you.
Wow, amazing. Can I get another round of applause for these guys? I think they were just absolutely amazing. So while we wait for a few questions to be roused from the audience, Amali, what inspired you to become a clinician and then a scientist?
Good question. In terms of becoming a clinician, I really loved science, and for me, I loved the aspects of medicine about being able to learn a lot of science, but then also to be able to work with patients and people to try and, I guess, explain that science, which was really appealing to me. And in terms of research, I got into research probably relatively later, in that I was working as a clinician for a little while. And a mentor of mine, Dr. Furlong, who's a kidney specialist, was starting a project here at Garvan about genetic testing in patients with PKD, and said, this seems like right up your alley, you should try this. And what I loved about research was that it allowed you to really deep dive into some of these conditions, and that it allows us to answer so many of the unanswered questions that come up in the clinic all the time, so that we can give better explanations to the people that are affected by these conditions.
Amazing, thank you. And forgive me, everyone, I should have prefaced the Q&A with the understanding that we can't provide individual clinical advice. Whilst these guys are clinician scientists, they can't provide you with advice on your particular conditions, but do feel free to come and have a chat with us at the end of the session. Owen, you were a little boy when you had your rare disease diagnosis. Is that what motivated you to become a clinician and a scientist?
I'm not sure it did, actually. So Amali's a clinician scientist. I'm a scientist clinician. So science was my first love, and I did my science degree first, and was really just enthralled by the whole genomics revolution. But during my scientific training, I started to see the research entering the clinic, and it was a really exciting time. The cost was coming down to the point at which you could really use this easily in the clinic. So at that stage, I saw a lot of my colleagues, like Amali, clinician scientists, who were doing this in real time, and I decided, no, I want to do that. So I did medicine later in life, and it was probably a number of factors, but I'd always been interested from a young age. I didn't know that disease was genetic at the time, but it was really seeing the research entering the clinic that inspired me to do the clinical training.
Amazing. Thank you for pointing that out. We've got a clinician scientist and a scientist clinician coming at it from both ends. Fantastic. Any questions from the audience? Don't be shy. Trent, we'll get a microphone to you.
Thank you, and thank you both for that wonderful presentation. To be able to give a talk about genetics that you can almost 100% understand is no small feat for a layperson. I had a question about something that you raised on, which was the newborn screening program, which has obviously been an essential tool for diagnosing in babies genetic diseases. I'm interested, we currently, I think, test for about, or screen for about 30 diseases. I'm interested in seeing what you think can and should be possible with newborn screening in the years ahead. For example, could we get to a stage where we do whole genome sequencing as part of the newborn screening program?
Yeah, absolutely. Very insightful question. So there's a lot of studies underway now in Australia and around the world that are testing this exact question. So for those of you who may not be familiar, the current standard is to get a heel prick blood test at birth, and then take that blood spot and then test it for a bunch of conditions, not using genetics, but using a different type of assay called sort of metabolomics. But there is a lot of discussion now about supplementing that, or potentially in the future, supplanting it with a genomic test, especially as the cost comes down further and further. So those studies which are underway now are going to address those points. There's one being led from Westmead Hospital at the moment, and actually we've just had a PhD student start in our lab this week who's going to be working on that project. But I think it's a difficult area, genetic screening, when you're testing before someone has a disease. So I'll hand over to Amali in a second. She deals with this a lot more than I do, but we need to be very careful about the information that we give back to families if we do find something that we think is potentially disease-causing. And we also need to understand, do the public want this, and how much do they want, and in what form? So A lot of studies underway, but a lot of people thinking about it.
Yeah, it's a great question, and I think really highlights the importance of research to try and answer it, because technically what you're saying is already possible today, that from a few tiny drops of blood, it certainly is technically possible to sequence someone's genome and get an incredible amount of information about different disease conditions that we know about today. But there are a lot of questions to be answered about whether that's something that we want to do and whether it's something that our current knowledge actually allows us to interpret that information. And I think that's one of the things that I really enjoy about working in genetics, is that the technology is really challenging us to improve our scientific knowledge all the time. And that though technically we can sequence a whole bunch of genes and get back a whole bunch of information about changes in those genes, our scientific knowledge may not quite be up to interpreting that in a meaningful way to a family so that it actually improves the quality of their care. And so those are the types of questions that are being looked at in research studies across Australia and across the world. And we're really privileged that Australia's actually a real leader in these kinds of questions and looking at them. And I think highlights some of the amazing things can happen in medical research when we can combine the technological advances with our scientific knowledge.
Thanks, guys. I'm just going to segue to a question from the Zoom room because it's related to what we've just been discussing, and then we'll come back to the individuals in the audience here in the auditorium. If a child has a genetic disease, has a genetic diagnosis, and they know that there are currently no clinical trial opportunities, what is the value to the family in joining your registry? Owen.
That's an excellent question. The potential is there. We really want to keep track of these people when they get a diagnosis. So in a lot of cases, we can test someone today, we can diagnose their disease today, but in cases like this, there might not be anything available. And what we really would hate to miss is losing that patient in the system. And if a treatment does come along in 5 years' time and a company really wants to enroll patients with a specific disease, we want to be able to find those patients as quickly as possible and as many of them as possible. So I think there is still definitely value in having patients participate in research, even if there's not an obvious immediate benefit. There can still be uses in, that this is medical research. We fill these gaps. So there may not be a direct benefit for you today as a patient, but your contributions, your information, your samples can potentially change another patient's life or be useful in the future. So I think it's definitely still important to engage in research.
Amali?
And I guess it also comes back to that, the power of numbers as well, in that rare disease for patients and families who have it can feel very isolating, and that, these conditions are super rare and you feel like I'm the only person in New South Wales that has this, or only a handful of people in Australia. But the value of a registry and putting information into there is that it allows us to identify those other handful of patients. And then even though there might not be a treatment or research today, a researcher might look at that and say, I didn't appreciate that there was all of these people with this same condition all grouped together. That is something that then we can focus on and make a meaningful difference. But when those people are all in separate states and different places, that information is unknown.
Brittany, you've got someone up the back?
Thank you. My apologies, I arrived a little bit late, so I didn't hear Owen. Maybe this has already been covered, but what I wanted to...
Hello, Charles.
Hi. Sorry, I'm a bit late. The question I had was in relation to the technology and how it's CRISPR technology in particular is being applied to understanding the correction of the coding and taking out faulty, replacing it with new, and then personalized medicine that will allow that replication to occur. Now, that's my simplicity in understanding. How far away are we from that?
So yeah, great point to raise. I didn't cover it in a lot of detail in my talk, so I'm glad to have the opportunity to now. So for those of you who may not be aware yet, there's a lot of these incredible gene editing tools available now. It was the subject of a Nobel Prize a couple of years ago. So exactly as was mentioned, these can specifically correct a genetic mistake. So they're perfectly suited to these types of diseases where we have a single genetic change causing a single disease. And already there are treatments like this approved in Australia, and patients are getting these right now. So, you know, one of the first of these types of medications approved was a gene therapy called Luxturna, and it's being used to treat blindness. And I see some of these patients occasionally in the clinics in Sydney. There's so many more of these technologies on the horizon, so both technologies and drugs on the horizon. And it's a really fast-moving technological field. So we're just going to see more and more of these. A lot of the trials are happening overseas just because, you know, Australia may not have enough patients. So that's another motivating factor for trying to corral as many of these individuals as possible because the treatments are coming.
Rory.
Look, given the old adage that prevention is better than cure, I'm thinking of, you know, you hear this term genetic predisposition, especially with psychiatric type conditions. I'm just wondering what you might have to say about that, because obviously if there's a predisposition and some environmental factor has to happen before it gets expression, that would be a good way to go.
Yeah, that's a great question, and I think it opens up to a whole kind of different field of genetics looking at risk factors. I think a lot of the conditions that Owen and I have spoken about today are what we call monogenic disease, where one very significant mistake in one particular gene results very reliably in a particular condition. And that accounts for a lot of the rare genetic disease. But we also know that genetics is really important in many more common conditions, that conditions like psychiatric conditions like diabetes, like hypertension, that we probably, or we do have very subtle genetic changes in lots of different genes that are combining in different ways with environmental factors to result in a particular condition. And there is lots of work being done trying to better understand those genetic risk factors so that you can so we can identify which patients are at highest risk of some of these conditions, and exactly as you say, then see if there are ways to modify some of the other non-genetic factors to reduce their risk. There's work done in different laboratories here at Garvan looking at that. Associate Professor Ingalls, who works in one of our cardiac disease laboratories, is looking at that specific question. I think Owen's done some work in eye disease on that as well.
But a work in progress, it's fair to say. Now, we've got a number of questions online that are specific to the registry. I want to whip through them really quickly, as many as I can, and then we'll come back and see if there are more questions in the audience. Does the registry include mitochondrial diseases?
Yes.
Excellent. Let's see. Does the registry link with other existing clinical or patient disease organisation registries?
Yes, so at this stage we've worked very closely with an organisation called Rare Voices Australia. So they're the leading rare disease patient advocacy group in the country, and they've been instrumental in shaping how this study has been put together. But through our partnership with Rare Voices, the intention is to then link directly, you know, if we can't provide the research service, to link out to other organisations and studies in the country that might be well suited to the particular patient groups.
Fantastic. Is enrolment in the registry entirely patient-led, or will clinicians be involved in recruitment?
Clinicians can and will be involved and have been involved, and we certainly rely upon clinicians providing the really important clinical information, which is important for us to understand the disease and the likelihood that it's going to be monogenic or caused by a single gene. So absolutely, we're designing it in such a way as to either the patient can provide their clinical information or upload a specialist letter, we're building ways for the clinicians to also directly contribute information as well.
And does the registry pull data from the health system?
Not yet, but that is at the very top of our wish list to be able to integrate with electronic health records.
And what would it take to do that? I'm going to ask you both this question. How do we better integrate research with the clinic?
Look, there's lots of different ways. There is a lot of paperwork, but yeah, look, I think that there's lots of ways through different layers of the health system, starting from individual clinics where you can collect patient information, which clinicians or patients could enter into registries, and then layers through kind of available information through Medicare benefits and PBS as well, which is kind of more big picture information on the health system. And there are some amazing research that's done through studies called data linkage, where they're able to link patients' access to the public health system with different health outcomes, and that's been a very powerful way to drive health research.
Owen, anything further to say?
No, absolutely. Yeah, there's so many fantastic resources at both the state level and the federal level, and that's still built into the registry design.
And what I can add from a whole of Garvan perspective is we are somewhat unique in that we were founded out of St Vincent's Hospital, founded by the Sisters of Charity. So that integration, certainly with St Vincent's, is in our DNA, if you'll allow me to say that. And that's a really important part of the way that we operate. And both Owen and Amali are a wonderful example of the importance of attracting young, talented people who can traverse both the clinic and the research laboratory. It's absolutely critical. I think we've probably got time for one more question from the audience. I'm sorry. And please do email through further questions following the event. Yes.
Yes, hi. I was diagnosed with a rare condition. The condition itself hasn't caused a lot of problems, but what it's linked to, various cancers, is of concern for me. But I went through Professor Alan Spiegelman, who did the genetic testing, and he's recommended screenings. But everybody has said to me, the professionals, it's so rare, don't worry about it. So I don't know how to view that. And would that testing that I now know I have, would he have put it into the register?
Absolutely not without your consent.
Well, it sounds something, and anyway, I know this is personal.
It sounds like you should reach out to the registry.
Yeah, no, absolutely. Nothing like that would happen without a patient's or participant's consent. So.
But is that unusual to be told not to worry because it's so rare?
We probably can't comment further on your personal case. I am sorry.
Okay.
So
can I make an appointment to talk to someone?
We could chat afterwards. Yeah.
Great. Thank you. Thank you. Look, everyone, I think this has been a fascinating session. Does everyone agree? Round of applause. Thank you so much for joining us. Thank you to everyone in the Zoom room. Absolutely wonderful to see you all. Hope that you'll be joining us for the next Garvan Public Seminar. We do sincerely value your feedback. Please let us know what you think. I believe you will get a follow-up email and a portal to do that. And if anybody would like to know more about the registry, or if there are any questions that weren't answered today, please send an email to foundation@garvan.org.au. If you have been inspired, please do consider making a donation to the Garvan Institute of Medical Research. And if you want to learn more about Garvan's research, I would warmly encourage you to Google the Garvan podcast series. It's called Medical Minds. We've got a wonderful recent episode featuring Owen, but there's lots of other great stuff there. So please do check it out. Have a wonderful day. Thank you.